But
Thank you. Yeah, sorry about this. There's some kind of privacy stuff that we have that I needed to disable, I guess, before I started in order to allow Zoom to have access to the
I see.
PowerPoint. It's
This should be
very,
fine.
that's,
Yeah. Again,
yeah,
just let
it's
me know in
frustrating. That's
advance.
actually why I sent the deck this morning of
That's
fear of this not working.
that's smart.
We've had this so many times, either external speakers have issues or we have issues. So
All right, looks like it's 7:01. So we have Rajita from Genentech who's going to talk to us about targeting IL-33 and TSLP. So thanks for doing this. And just FYI to everyone, I'll be advancing the slides, so I apologize in advance. But this should go just fine. So thanks for doing this talk for us.
Yeah, good morning, everyone. My name is Rajita Pappu. I'm a scientist at Genentech in the Immunology Discovery Group. I've actually visited you guys before. So Dr. Altman, I came and gave a talk on IL-33 like about 6 or so years ago. And today I'm going to be discussing a review article that we wrote about IL-33 and TSLP, or thymic stromal lymphopoietin, and about in the basically summarizing what's been published on their biology, genetics, and clinical data. So could you, and my colleague Divya Mohan is also on the line, and she'll be available to answer any questions about any clinical data. She's from the clinical group. Please, next slide, please. So we appreciate, damages to the lung epithelial barrier result in a lot of inflammatory cascades that ultimately lead to what we see clinically in asthma and COPD, which are decreases in FEV1, increased rates of exacerbation, and increased symptoms. Next slide, please, or advance, please. And so really the goal of the current therapeutic landscape is to try to develop drugs that can reduce the exacerbation rate, and that's the primary endpoint for these diseases, and improve FEV1 as well as symptoms, and in the case of asthma, reduce oral corticosteroid use. Next slide, please. So we appreciate that these pro-inflammatory signals that come from the perturbations at the epithelial barrier that are caused by allergens, pollutants, microbes, etc., lead, really can activate on a number of cell types that we think are involved in asthma and COPD exacerbations and changes in FEV1, etc. So these include sensory neurons that are involved in the cough, airway smooth muscle involved in bronchoconstriction, and there's of course the crosstalk between the neurons and the smooth muscle. Goblet cells that lead to excess mucus production, and then fibroblast activation that can lead to structural changes within the lung tissue. Next slide, please. And so today I'll be discussing with you the biology and some clinical data that support a role for alarmins in propagating these inflammatory cascades in asthma and possibly also in COPD. And so TSLP and IL-33 they're considered alarmins because they're rapidly released upon epithelial damage. And I'll walk through the biology of each of these cytokines and then some of the genetics and clinical data that support a role in these pathogenic processes. So next slide, please. So first I'll talk about TSLP, and please feel free to stop and we can discuss as we go along. This is a member of the IL-2 family of cytokines, and it has a shared receptor with IL-7. And so the specificity of its receptor, as you can see on this target cell, which generically highlighted here, is through expression of TSLP receptor. And so one of the unique things about TSLP is that it's transcriptionally regulated. And so in response to a viral, bacterial allergen, or even a pro-inflammatory cytokine stimulation, epithelial cells, fibroblasts, and even immune cells, including dendritic cells and mast cells, are then turned on to transcribe TSLP and secrete TSLP as part of this reaction to the stimuli. Next slide, please. So TSLP has a property of activating both the innate and adaptive immune responses, and hence, as we go through some of the clinical data, might be why it's been so effective, at least in asthma, in reducing exacerbations. So one of the key target cells you can see here on the left for TSLP are dendritic cells. And through activation of dendritic cells, the TSLP can augment their antigen presentation capability, leading to differentiation of the CD4 positive Th0 helper cell into either a Th2 cell, which can then lead to B cell IgE production, mast cell degranulation, and production of pro-inflammatory cytokines such as IL-4 and 13. But TSLP can also lead to the differentiation of Th17 cells, which we know are important for IL-17 production and neutrophil mobilization. And so you can appreciate that because of its role in dendritic cell activity, that TSLP can really shape the adaptive immune response to either a type 2 or a type 17 reaction. And another feature of TSLP biology is its ability to activate innate cells. And so we've highlighted here these ILC2s. They're the innate lymphoid cells that behave very similarly to Th2 cells, but because they're innate, they respond very quickly and don't require any differentiation process. And they're been described to generate copious amounts of type 2 cytokines, including IL-5, which can drive eosinophil mobilization and activation, and IL-13, which we appreciate has a role in airway smooth muscle and goblet cells, as well as fibroblasts. And so you can appreciate here that TSLP, as this alarmin that gets released in rapid response to any external stimuli, can drive both adaptive and innate immune responses, so the type 2 high as well as non-type 2. And we think that this is really important for all inflammation as well as possibly tissue remodeling. So next slide, please. And so there's been several genetic studies that have implicated TSLP as a risk factor for asthma, developing asthma. And so this is just one such publication. You can see the Manhattan plot here that identified a SNP in TSLP that associated with asthma risk. And I put the red arrow to show where the TSLP is. And you can see the signal is quite strong, and it's only really second to the MHC signal in asthma susceptibility. And there are many other studies that have also described this genetic association with asthma risk. Next slide, please. Okay, so now I'd like to just talk about, a lot of the cellular and the genetic data have pointed to TSLP as having a role in propagating signals that we think are really important for airway inflammation and activation of these cell types, which we've shown at the bottom, that lead to the pathology. And so naturally, there's been a lot of efforts in the pharmaceutical industry to try to block TSLP activity. And you can see the antibody here, it's to the cytokine. So this is AstraZeneca's Tezepeluma. And I'll share a little bit of the data and we can discuss some of the findings too. So this was approved as an add-on maintenance treatment in severe asthma that was irrespective of eosinophil status. And the primary endpoint was in reducing exacerbations. In another study, they also demonstrated that it was effective in inhibiting mannitol-induced airway responsiveness, which is not targeted by the eosinophil-directed biologics like anti-IL-5s or IL-4/13. And so, as I mentioned, this effect on exacerbations and treatment effect that was irrespective of the eosinophil status really points to this MOA that we appreciate that has both the type 2 effects as well as the non-type 2 effects. And currently there is a phase 2 trial to test the effective TSLP blockade in COPD, and we've captured the clinical trials number at the bottom. And furthermore, Novartis is developing an inhaled formulation of TSLP in which they are testing this in both asthma and COPD to see whether a local delivery of TSLP is sufficient to see the same effects clinically on reducing inflammation and ultimately improving patient outcome. So the next slide, please. I thought I'd just capture one piece of really remarkable data from the TSLP phase 3 trial. So this is from the Navigator trial, and the references at the bottom. And this was their examination of looking at reduction of exacerbations with anti-TSLP blockade. And you can see here on the very left side, they're looking at overall exacerbations, so all comers. And this is stratified gray is placebo, and pink is with the anti-TSLP. And you can appreciate this reduction in overall exacerbations with the TSLP blockade. And then they go on to stratify based on eosinophil counts, so marking who is type 2 low versus type 2 high. And they still saw effects in the type 2 low, so the less than 150 cells per microliter, so eosinophils. But what was really remarkable was the effect that they saw in the type 2 high on the very right, where you look at the greater than 300 cells per microliter. And again, this really points to what we think about TSLP biology, that it has this role in the adaptive immune response, right, through dendritic cells and promoting type CD4 positive Th2 cell activity, but also in the innate cells like ILC2s and mast cells that can contribute to eosinophil mobilization, IL-4 and 13 production, et cetera. And so next, this was really, the next slide is also that they had an effect on improving FEV1. If you look at the Tezepeluma data at the right with the red versus placebo. And we know from other trials that FEV1 is driven a lot by IL-13 and the type 2 axis. And so it's not surprising that they saw an effect in this endpoint as well. Next slide, please. And so, one of the goals obviously in asthma treatment is to try to reduce oral corticosteroid use. So there was a trial with testing anti-TSLP on this parameter, and it really stemmed from some of the biology that suggested that type 2 inflammation, if we blocked it, there was then a concomitant relief of steroid effects. So what they did here is they did a 48-week study with Tezepeluma looking for, at the end, primary endpoint, a reduction from daily oral corticosteroid use. And although the trial primary endpoint was not met, if you looked at the cumulative odds ratio in the greater than 300 cells per microliter eosinophil, so type 2 high population, you can start seeing that there was a reduction of OCS use, that they showed the better response. And I think, this is still obviously work in progress to try to understand the mechanism behind TSLP blockade and reduction of OCS. So I think further clinical investigation will be needed to better understand this response and see if it really holds in this type 2 high population. So next slide, please. So just before I get into IL-33, I'd like to just summarize, and we can discuss now or at the end after we go through the IL-33 biology, that, Tezepeluma, one of the incredible parts of this alarmin is that it does have such a broad mechanism of action, hitting both that adaptive and innate immune response. And really, the clinical data suggests to us, at least in the human setting, that this is a primary driver of the type 2 biology, in that they saw a really great effect in reducing exacerbations, irrespective of eosinophil status, but the best benefit was in those EOS high population. And there's also some data that this population could also benefit with reducing their steroid use with this treatment. But of course, it's still early stages. We need to understand, does TSLP blockade also lead to disease modifying effects? And the role in COPD is still unclear. So, we'll have to just wait for that clinical data and see whether, similar to asthma, if exacerbation rates are reduced with anti-TSLP blockade. And that is if you assume that the triggers for exacerbation are similar across the two diseases. So next slide, please. So now I'll talk about IL-33. And I actually, just before I start, my lab has worked on IL-33 for many years. And so we contributed quite a bit to both the cellular biology as well as the genetic association, which I'll share with you. And our interest in, of course, moving IL-33 blockade into the clinic. So this is like TSLP, considered an alarmin. It's an IL-1 cytokine family member. So it actually shares its receptor with all IL-1 family. So the IL-1 RACP is a shared receptor across that cytokine family. And specificity here is dictated by the IL-33 receptor, which we call ST2. And the gene for ST2 is IL-1 RL1. And so there are two forms of ST2. One is the membrane form that is important for propagating the IL-33 signal on target cells. And the other is an alternatively spliced form, soluble ST2, that we think is a negative regulator of the pathway. So it acts as a sink to block endogenous IL-33 activity. So the difference between, one of the key differences in biology between IL-33 and TSLP is how they're regulated. So as I mentioned earlier, TSLP is transcriptionally regulated in response to any environmental challenge. However, IL-33 in contrast is constitutively expressed in the nuclei of cells, including airway epithelial cells, fibroblasts, and endothelial cells. And so it's only upon cellular damage, which you can imagine is a pathogen or a cigarette smoke or a pollutant or just even a mechanical stimulus, can then lead to the cell having a damage signal and then releasing IL-33 to allow for bioactivity. And so in this way, it's very different from TSLP. If we think of this as a very rapid response, and it's irrespective of the type of signal. So anything that will cause cellular damage, you can imagine, will lead to IL-33 release. It can also be induced in certain cell types like macrophages under inflammation. And so some of the triggers for IL-33 release and cellular damage include viral and bacterial stimuli, allergens, and pollutants. And so this is, as all other IL-1 family members, IL-33 signals through the MyD88 pathway to promote inflammation. And so there's really no specificity in terms of once the signal is started in terms of an IL-33 signature, because it is shared with all the other IL-1 family members. So really specificity is purely dictated by ST2 expression on the target cell. Next slide, please. Okay. So similar to TSLP, we appreciate that IL-33 has multiple target cells and can shape both type 2 and type 1 immunity. And so first, for type 2 immunity, it is a potent activator. You can see these ILC2 cells, as well as CD4 positive T cells in inducing type 2 immune cytokines like IL-4, 5, and 13, and allowing for eosinophil activity. Similarly, IL-33 can activate mast cells, and we've actually found that mast cell activity through IL-33 is not necessarily type 2 immune responses. We see non-type 2 cytokines as well when you stimulate mast cells with IL-33. But what's very interesting with IL-33 biology and ST2 regulation is that it's been shown that agents like cigarette smoke can actually shift ST2 expression to different cell types. And so what was shown was that when you give cigarette smoke, the type 2 cells downregulate ST2 expression, and now you have upregulation of ST2 expression on non-type 2 cells, such as CD4 positive Th1 cells, NK cells, and macrophages. And if you think in terms of the inflammatory response, when you have this shift of ST2 expression and you have cigarette smoke that then leads to IL-33 release, you can now have induction of non-type 2 pathways, including expression of interferon gamma and IL-12 and mobilization of neutrophils, which we appreciate, especially in COPD, has pathogenic role in lung damage. One of the other features of IL-33 is that's been coming up more in the literature is its regulation of hematopoiesis. And particularly, it's been shown that it can regulate eosinophil development. And so it may have roles in eosinophil biology independent of IL-5, which is still an area of active investigation. So next slide, please. So like TSLP, there's been many studies that have revealed a genetic association of IL-33 with asthma and COPD risk. So loss of function mutations in IL-33 are associated with reduced risk of developing asthma and COPD. And similarly, gain of function mutations increase this risk. And we have, and others have identified SNPs in both IL-33 and the IL-33 receptor. IL-1 RL1 is the gene for ST2 that have been identified to associate with asthma susceptibility. And we've shown that protective SNPs in IL-1 RL1 lead to reduced IL-33 signaling and increased soluble ST2. And I'll share some of that data in the next slide. Next slide, please. Okay, yeah, so then I just wanted to show where the SNPs, so they affect the ST2 membrane as well as the soluble form, and we'll get into a little bit of that in the next slide. Okay, so there was an original paper in New England Journal of Medicine shown here on the left by Moffitt et al. That first showed the genetic association of IL-33 pathway with asthma risk. And here what they found was SNPs in both IL-33 and the IL-33 receptor, which IL-1 RL1, that associated with asthma risk. And our group went on to examine these polymorphisms in our own data sets, trying to see whether we could stratify between the type 2 high and type 2 low to tell us a little bit more of where IL-33 may be having a role in vivo. And what we found was that these polymorphisms associated with asthma risk, regardless of the type 2 status, you can see here in the pink and the green, the odds ratios are very similar. And this is in contrast to other Th2 genes, which when we did the analysis, we found they were associated with a greater risk in type 2 high biomarker group, such as GATA3. And so next slide, please. So when we did this analysis, we also identified SNPs that led to missense variants, that led to changes in amino acids that were in the ST2 cytoplasmic tail. And so this, well, we found that this haplotype, they were in tight linkage disequilibrium with the original SNP identified in the Moffitt paper. So we found that this haplotype, if you look at the odds ratios, is protective for asthma risk. And because they were amino acid mutations, we thought, okay, let's see if in the cytoplasmic tail of the ST2 receptor, let's see, well, maybe this alters IL-33 activity, and can we test this in a cell-based assay? And so next slide, please. And so what we did is we identified individuals that carried either the protective haplotype, where they have the 4 amino acid mutations in the ST2 receptor, or the common haplotype. And we asked, well, do the eosinophils from these individuals have a different altered IL-33 activity, at least in a cell-based assay? And so here we isolated eosinophils from peripheral blood from these individuals and stimulated with IL-33 and measured activity by the release of IL-8. And you can see here common in red and the protective haplotype in blue, that individuals carrying this protective haplotype have a reduced activity in response to IL-33, as seen by IL-8 production, compared to those with the common haplotype. And similarly to the reduced IL-33 signaling, we also found that these individuals with the protective haplotype had increased expression both at the RNA and protein level of the soluble ST2 receptor, which we think is the negative regulator of the pathway of the sink. And so this was really exciting to us because it was very much in line with the hypothesis just based on the genetic analysis. Next slide, please. And so this is, we are, one group that investigated this, and there's many other reports about IL-33 genetic association with asthma and COPD. And this is a paper I pulled from the Lancet Respiratory Medicine, where they described individuals that had a GG allele in IL-33 versus the GC allele that associated, it was a loss of function variant that they found led to reduced levels of IL-33 in the serum, and the loss of function variant is shown as the GC allele. And what they found was that if you just look at C and D, comparing the number of cases versus controls in the individuals with GG versus GC, you can see those with the GC allele there's fewer cases for asthma and for COPD, suggesting that reduced IL-33 expression in the serum is somehow protecting these individuals from developing this disease. So next slide, please. Okay, so the genetics and a lot of the cellular biology really support investigating therapeutic blockade of IL-33 or anti-ST2, the receptor, in asthma and COPD. And so there's been a number of companies that have taken this approach. Regeneron, they have a program against IL-33 cytokine, itepekimab, and I'll go through some of the data that at least in one of their trials, it failed to demonstrate superiority to IL-4/13 blockade through dupilumab. AstraZeneca has a program also targeting the cytokine for asthma underway. And I'll share some of the data on our own program, astegolimab, anti-ST2, which had effect on reducing exacerbations in the type 2 low population. And GSK has discontinued their anti-ST2 due to low efficacy in their asthma trial. But one of the things, we assume when we start these kind of trials is that you're going to see a similar effect between blocking the ligand versus blocking the receptor. And I think the more we, analyze this data that's coming out clinically, and the more we understand about IL-33 and the different forms of IL-33, it would really help us understand what really is the best target to block this pathway. Next slide, please. So this was just capturing our astegolimab anti-ST2 trial, Zenyatta, in severe asthma patients. And you can see here that we had a placebo arm and then 3 different doses of astegolimab, including EOS low individuals as well as EOS high individuals. And the primary endpoint was the reduced annual asthma exacerbation rate. And what you can see here is that overall, we did see effects in all the all-comer population in reducing exacerbation the 52-week time point. But really where we saw a greater effect was there, you can see at the 490 dosage in the EOS low population. And so we think, and I'll go back to what we think about IL-33 biology, but we think that this also reflects the fact that IL-33 has a effect on this non-Th2 population, and this could be why we're seeing effects in this group. Next slide, please. So around the same time we were, examining anti-ST2, Regeneron also tested their anti-IL-33, so to the cytokine, not to the receptor. And their trial was different from ours. The endpoint was not exacerbation, it was the loss of asthma control. So we can't really compare them. But what they did here is that they looked to see whether there was an effect that on top of dupilumab or unique from dupilumab. And what you can see here at the bottom is that the IL-33 blockade was very similar to dupilumab, and then there was no greater effect when you combine them together. It was just slightly, you can see the blue versus the red and the green. But it is a different trial design, different endpoint, so it's hard to compare the role of IL-33 in driving this endpoint versus driving the exacerbation endpoint. Next slide, please. Okay, so there's, we think because IL-33 is an alarmin and it's released upon cellular damage, which could come in, as an allergen, but it could also come as cigarette smoke, right? Or a pollutant. So we think that it could have a role in, non-traditional type 2 responses, which we think of in allergy. And so there's been a lot of interest, obviously, because of this hypothesis in testing IL-33 pathway blockade in COPD. And so, of course, Regeneron has a trial which they published, a phase 2A, and as well as a phase 3 that's underway. And we also have a phase 2A that was published, and then we have a phase 2B underway, trying to see whether the hypothesis, does IL-33 have a role in a more mixed granulocytic disease like COPD and reducing exacerbations. So next slide, please. So this is data from the Regeneron's trial blocking the anti-IL-33 cytokine in COPD, and the reference it's from the Lancet Respiratory Medicine. They had about 170 patients per arm. And if you look here in B, that's their annual exacerbation rate at 52 weeks. You don't really see a difference between the placebo and the IL-33 blockade. But when they stratify in D, looking at former smokers' response to IL-33 blockade, that's where they start seeing clinical effect. And that IL-33 blockade appeared to have reduced exacerbation rate in former smokers, but in F, you can see there's no change in current smokers. And so it's, still early. This was a phase 2 trial. And I think trying to understand the effect on the smoking population is still an area of investigation. So next slide, please. Okay, and so this is just a high level from our phase 2 trial. It was very small, 40 patients per arm. But we also had the hypothesis that if IL-33 is indeed released upon any type of cellular damage, it may have a role in a pollutant, based disease like COPD. And so what you can see here in B is the placebo in orange and the astegolimab treated group in blue, that we didn't see a statistically significant reduction in exacerbations, but a trend in reduction, especially in that less than 170 cells per microliter type 2 low population. And so that was very similar to, of course, what we observed in the Zenyatta trial, where we saw the greatest effect of astegolimab treatment in the type 2 low population in reducing exacerbations. So next slide, please. And so this is actually like my final biology slide, and we can open it up. I'd be happy to have further discussion on the data. But the asthma studies really highlight an important role for these alarmins, TSLP and IL-33, in driving pathology that we've captured here that ultimately leads to the exacerbations and the FEV1 reduction. And anti-TSLP appears to have a really strong effect in driving type 2 immune responses. And we think this biologically could be that because it has a role in controlling adaptive as well as innate immunity. Whereas IL-33 appeared to have a greater effect at the type 2 low. And it's hard to understand, IL-33 didn't have as great an effect in type 2 high, and it could be redundancy with other pathways like TSLP, or we just other pathways instead of TSLP are also contributing to induction of type 2 high immunity, even in the presence of IL-33. And one of the key, I think, interesting as we learn more from clinical data is going to be around, in IL-33 case, is there are going to be differences between when we block a ligand versus we block the receptor. So there's a lot of literature out that are describing different isoforms of IL-33, and that there's an oxidized form, there's truncated forms, et cetera. And it's really not clear whether if you block one versus another isoform clinically, if there's going to be a different effect. And I think as more data becomes available, we'll be able to understand that distinction more carefully. And finally, there's a lot of momentum with the COPD trials in blocking both TSLP and IL-33. And one of the key questions obviously will be around safety as we get more clinical data. So far, there's no description of an imbalance of safety events. but the risk of infections will only be evident over time. And also the secondary endpoints, like an improvement in FEV1 and symptoms, is also yet to be determined. And so with that, the last slide, I have to tell you, the word of the day is cosmos. Now I'll open it up to any questions and discussion and comments. And Divya's on the line also, if anybody has clinical questions that she can chime in.
Thank you so much. Yeah, we'll open it up here.
This is Vinod Doraiswamy, one of the local allergists. Thank you for a wonderful talk. Certainly new information. Certainly trying to get the head wrapped around, trying to understand everything. But what do you see? I had two questions. One is, when you start blocking, of course, with dupilumab, now you start, kind of seeing some issues with, say, psoriasis or enthesopathy. What do we think about blocking IL-33, and what other avenues or other potential side tracks or inflammation do we expect to see, by blocking that IL-33? And second part is, what do we see the role of multiple biologics, not, cost aside, and, in terms of management? Thank you.
Yeah. Divya, do you want to take this one, or do you want me to, this safety question?
I can take a part of it, and you can also comment on the biology. I think as far as IL-33 in skin diseases, I know that AstraZeneca have got ongoing studies in atopic dermatitis. We did our own. That was negative for all clinical endpoints showing any improvement. So at least, again, in the type 2 inflammatory pathways, doesn't appear that IL-33 has that effect. Other diseases such as psoriasis, etc., haven't seen any reports of that. So I don't know, Rajita, if you can comment on the biology, but I'll just quickly cover the other question on multiple biologics. And I think the respiratory space is still early, in that regard. And I just wonder if we will see more switching of biologics over time, kind of what's occurring in rheumatoid arthritis and the rheumatological diseases. So I don't think there's going to be one biologic to cover everything. But if patients evolve over time, I think we need more data to understand whether they may benefit from a switch. Sorry, go ahead, Rajita.
Yeah. Oh, yeah, no problem. So I'll comment on both also. So in terms of skin, we haven't seen, like Divya commented, we have not seen, real huge role for IL-33, at least in activity in any of the human skin disease tissues we've looked at. And like she said, we did run the AD trial, and I think the lack of efficacy there was probably that. Going back to this redundancy question, there's many ways to get to IL-4 and 13. There's IL-33, there's TSLP, and I guess depending on the trigger that's leading to the disease, you'll have, release of one versus another. And then obviously if there's redundancy, it's better to block downstream than upstream, right? And then in terms of combining biologics, if you do preclinical studies in mouse, yeah, we absolutely think that it's great to combine, because you can hit this pathway and this pathway all, and get it all in one shot. I guess ultimately it comes down to, are we going to see greater safety issues, right? If you try to combine biologics or try to make a bispecific, trying to target two different cytokines or two different pathways. And, so I think, as Divya said, as we understand disease better and the biomarker better, and, which patients are responding to what targeted therapy better, we'll be able to make that, address this question of the multiple biologics, or at least, bispecific type approaches, or trying to use a small molecule inhibitor that may go after multiple cytokine pathways. You know, there's a lot of interest in going after JAKs, obviously, because they can control different type 2 cytokine pathway. So I think we just need to, you know, understand the human response data and the response better before we, you know, go down those paths. Other questions or comments?
You know, this is Vinod here again. I'm sorry. You know, the first question was actually pertaining to safety risk in terms of blocking IL-33. Do you have other downstream effects or risks by blocking IL-33, not necessarily role of IL-33 in atopic dermatitis or psoriasis? It's just that what's the risk of blocking IL-33 and what are the other things seen in clinical trials so far? Thank you.
Do you want me to take that one, Rajita? So,
Yeah, go ahead, please.
Sure. So in terms of safety, at least the experience with our molecule, we've now conducted about 4 studies with astegolimab in the clinic and have data for over 500 patients. So at least early on, we don't actually see any imbalance in safety events to date. So we haven't seen an increased risk of infection. We've not seen imbalance in cardiovascular safety, which is described as at least in preclinical literature as a potential risk. So far, it actually looks pretty clean. Now, of course, as you get more data, you know, the phase 3 studies going on now, so we will get data from thousands of patients. So I don't know if something else will emerge, but certainly the only preclinical risks that I've seen described, I think one relating to type 2 biology that, you know, is there an increased risk of helminthic infection? We haven't really seen that in the clinic. And I think the other risk, as I mentioned, is a cardiovascular risk preclinically. Again, haven't seen that in the clinic.
Any other questions or comments?
All right, if there's no more questions, I think we can let people go a little early. Thank you so much for an excellent talk. We appreciate it.
All right, thank you for your time. See you.